Thank you very much. Dear presidents, dear colleagues, it's a great honor to be here and to present recent knowledge and our results related to the role of damage-associated molecular patterns in chronic renal injury and inflammation. As we know, chronic renal injury represents one worldwide health problem recognized as a common condition associated with the increased risk for atherosclerosis and related, presumably, cardiovascular complications. We know that there are many causes which can induce chronic renal failure. Among them are diabetes, hypertension, consumption of alcohol, infective agents, smoking, etc. But as we talk about these factors, we can divide them into modifiable and not modifiable. We know that not modifiable are age, sex, gender, ethnicity, but among the modifiable are, as I already told about them, diabetes, increased protein intake, smoking, dyslipidemia, and other. When we have first kidney damage, then many compensatory mechanisms may be involved to solve this problem, to remodel tissue. But among them, the first of them is, as a defense mechanism, is inflammation. It acts as a defense mechanism, but because if it persists, they can induce very serious kidney damage. When we talk about different types of agents capable of inducing kidney damage, we can divide them into pathogen-associated, known as PAM molecules, which represents a large group of microbial agents, and then these particles are essential for their survival in the host. You know about, for example, lipopolysaccharide. But when we talk about the damage-associated molecular patterns, we can see they represent multifunctional host molecules which are released from damaged cells, or they can be secreted from damaged cells during, if cells are so healthy, they can secrete them. But if they're necrotic, then they are released into extracellular space. We have up to now identified a lot of damage-associated molecular patterns. You can see here that among them are very recognizing nucleic acids, ATP, adenosine, galactins, thioredoxin, S100 proteins, high mobility group protein 1, then another, you can see here a number of them. Now we can see that from one side we have strangers, they are pathogen-associated molecular patterns. From the other side, we have dangers, which are released or secreted from damaged cells. But for them, for both of them, is typical danger hypothesis, which postulates that the immune system reacts to both almost equally, inducing inflammatory reaction. Are they strange molecules or derived from our cells? And they can induce, damp molecules can induce presentation, activation of antigen-presenting cells, and they are capable of inducing secretion, strong inflammatory response and secretion of inflammatory cytokines. Among them, the first line is TNF-alpha, interleukin-1, then interleukin-6, 8, and also the expression of cell adhesion molecules. Inflammation is strictly related to the activation of innate immune cells. Among them, we know that normally we have in kidney tissue macrophagal cells, but during damage they can be activated to be M1 type, which is capable of secreting a number of cytokines, and it is capable of inducing strong inflammatory response, which can lead in some instances to the fibrosis, but also they can go to the another state. It is M2 phase, which can induce repair, but it is also responsible for fibrosis. Are damp molecules only bad guys? From one instance, they can be good influences of tissue remodeling, but in other side, beside the inflammation, they can induce platelet granulocyte interaction, then platelet activation, and both of them can result in increase of renal injury. Maybe the question is, what are criteria to establish some substance to be damp molecule? First of them, it should be active when it is highly purified. The second is that this biological activity is not related to any contamination with some PAMS molecules with pathogen associated. They should be active in both physiological concentration and especially active dose-dependent effect that should be noticed when they are purified. Also, they can be inhibited by specific antibodies or by specific microRNAs. The second question is, what recognizes damp or PAM molecules at the cell? Up to now, we can register a number of receptors capable of binding these ligands. First of them, maybe the most important, are toll-like receptors. We have a number of toll-like receptors, then RAGE receptors, and node-like receptors. Toll-like receptors are the central class of pattern recognition receptors, and they are located. Why they are interesting? Because they are located not only in immune cells, but in all tissues, and their expression depends on the extracellular environment. We will see later about mesangial cells, for example. Excessive activation of toll-like receptors can induce strong inflammatory response. We can see here that macrophagal cells and neutrophils are very rich in toll-like receptors, and their activation by number of downstream signaling pathways can induce secretion of cell adhesion molecules, a number of cytokines, number of chemokines. The other cells very rich in toll-like receptors are endothelial cells, which can also capable upon stimulation of toll-like receptor to secrete cell adhesion molecules, cytokines, and chemokines. As concerns the mesangial cells, they normally have a number of toll-like receptors, but in the we can see here that in the situation when we have PAMP-associated molecular patterns or DAMP, almost we have a similar signal transduction pathway. What are the ligands for DAMP molecules of RAGE receptors? First of them, the name they receive, because firstly it was recognized, discovered that they can bind glycated proteins in diabetes, then DNA, RNA, high mobility group box protein 1, advanced oxidation protein products, glycated LDL, S100 proteins, amyloid, and heat shock proteins. All of them can activate the specific pathway, and they can activate transcription factor NF-kappa B. What is also important to say that DAMP molecules induce sterile inflammation, and then during this sterile inflammation they can attract a number of neutrophils which are capable of secreting free oxygen radicals, reactive oxygen species, and it is further tissue damage. They can also induce microvascular congestion, which can induce impaired blood flow, and there are many criteria to establish some kidney damage in this situation. What about NF-kappa B? It is the main transcription factor responsible as acting as a bridge between different ligands, whether they are PAMP or DAMP molecules, to the DNA molecule, to the promoter sites of DNA. And we can see that the activation of NF-kappa B can induce activation of pro-inflammatory genes, can induce either apoptosis or proliferation, depends on the state of the cell, and maybe in some instances pro-fibrotic genes. Normally, NF-kappa B is present in the cytoplasmic site of the cell, but bound to its inhibitor, and then after release of its inhibitor, which is immediately degraded, heterodimer P50 and P65 translocated immediately to the nucleus, and then it binds specific promoter sites for induction of a number of cytokines, toll-like receptors, growth factors, chemokines, apoptotic regulators, adhesion molecules, and also can induce itself subunits. Since we saw that NF-kappa B is so important in inducing inflammatory response, a number of approaches has been documented in literature to decrease the activity of NF-kappa B to find suitable inhibitors. Now we will focus about circulating nucleic acids, because we saw that they are equally active by binding to the toll-like receptors and RAGE receptors. The binding for toll-like receptors is strictly specific because toll-like receptors 7 and 8 can bind single-stranded forms, 12-3 double-stranded, and toll-9 can recognize ligand unmethylated CpG. It is part of DNA. It is very important to note that whenever their nucleic acid can be exogenous, derived from some viruses, from some bacteria, or endogenously generated, they can induce inflammatory response. And the second very important part is that they can they can induce normal cells to become antigen-presenting cells. We have one example concerning the mesangial cells, which, for example, do not express toll-like receptors 3, 9, which are capable of recognizing nucleic acid. But when they're in an environment where they have nucleic acid in extracellular space, then the cells immediately express toll-like receptors, and then they immediately induce inflammatory response. The inflammatory response is characterized by the strong secretion of pro-inflammatory cytokines. It is very known that those toll-like receptors 3, 7, 8, and 9 are capable of different nucleic acids and can induce strong inflammatory response. What are the other effects? Besides the secretion through the NF-kappa B, we have secretion of cytokines, inflammatory response, but nucleic acid can also act by binding with MDA5. It is very important transcription factors, and then they can activate transcription factor interferon-related factor 3, 5, 7, and all of them are capable of activating interferon secretion. We can ask, are there any way to have some gatekeepers to protect the extracellular environment against nucleic acid? We are occupied so much with this problem because we are measuring activity of a number of DNases, RNases. I hope in next time you will be introduced with our research related to the effect of these gatekeepers. But now I will shortly introduce you in our study where we first determined NF-kappa B level, P65 units, P38, MDM5, IRF3, IRF7, and then this was estimated in patients, not in patients, but in the nucleic acids are isolated from plasma of patients in different stages of chronic renal failure, and also we have patients on dialysis program, including in stage 5, and of transplanted patients. Nucleic acids were isolated according to the standard procedure. Culture of resident macrophagal cells was established, and then macrophagal cells were treated with different nucleic acid species, isolated. We can see here that some clinical characteristics of patients, urea level, creatinine level, and now you can see concentration of circulating nucleic acid, and we can see here that the highest level was in chronic renal failure 4th and 5, but in transplantation it was a little bit lower. When we studied spectrophotometric scan analysis of these nucleic acids, we found that they have a different concentration, but similar characteristics, because peak of their concentration was at 270 nanometers, which correspond to the policy. They are very rich in cytosine. And now we can see shortly our results. The activity, the level of NF-kappa B has been significantly increased, almost 2 times, in patients in macrophage cells treated with RNA of patients in 4th and 5th stage, while if macrophage cells are treated with control samples where RNA has been isolated from controls, healthy controls, we can see decrease. What about IRF7? As concerns the IRF7, we receive increase in healthy persons, but decrease in patients in renal failure. Since we know that IRF7 is capable of inducing secretion on interferons, we can postulate, we can hypothesize that it can be the reason why the patients with chronic renal failure are more susceptible to viral infections. They have no activation of IRF7, but we receive a strong increase in patients, in transplanted patients. At first sight, we thought it is very good, but when we found some literature data, we found that patients have been already tried to be treated with interferons, but the effect was not as can be expected because they induce some humoral rejection of allotransplant of these patients. The similar situation has documented for interferon-related factor 3, which is presumably responsible for secretion of interferon beta, while IRF7, we saw that it is responsible for secretion of interferon alpha. What about MDA5? It is also very important transcription factor capable of recognizing RNA molecules, but we saw that it was down-regulated, it was decreased in all macrophagal cells in all cultures. What about P38? We can see that P38 is an important part of mitogen-activated protein kinases responsible for some proliferation, cancerogenic effect, has been without effect. Only in transplanted patients it was decreased. Then we can talk something a little bit about the results in literature. We found that presumably immunohistochemical analysis have shown in bioptic materials for patients that in IgA nephropathy, in lupus nephropathy, in diabetic nephropathy, that NF-kappa B has been highly expressed. Then there are some maybe we can say drug repurposing effects, because some patients have been treated due to, for example, other diseases such as multiple myeloma. They have been treated with NF-kappa B inhibitors. One of them is bortezomib. And they found that they observed rapid improvement of renal function. What can explain in some instances that NF-kappa B inhibition can be successful in ameliorating renal damage. What about experimental models? There are many experimental models where different NF-kappa B inhibitors can be used, then microRNAs and some other substances. Also, we have found some results about some Chinese plants, and in our conclusion is that obtained results may indicate that circulating nucleic acid can act as a damage active molecules, and then they can induce strong inflammatory response via activation of transcription factor NF-kappa-B, while the effect through the interferon-related factor 3, 7, and MDM5 was without effects. Thank you for your